Hey, what's up? This is Coach Hyde. What's your name?
Rowan.
Rowan. This is Roro, my little girl. All right, so today we're doing the starch, the yeast sphere experiment. All right. Cool. Okay. We are studying a protein. All right, an enzyme specifically. Proteins can be a lot of things. They can be enzymes. So we're studying enzyme function of an enzyme called catalase. Now, catalase is an enzyme found in our liver. It's also found in yeast. It can break down hydrogen peroxide. So what we're focused on, like we talked about the other day, careful, is that enzyme function can be changed, right? Enzymes can be denatured. So depending on the environment that the enzyme or the protein is in, it may denature, right? So if we have a fever, it could denature the protein, it won't work as well. So we're studying some different conditions and how this affects the activity of the enzyme. We're going to have a control group. This is just starch balls, right? Just starch spheres and water. It's nothing out of the ordinary, right? We didn't do nothing to it. And then these other colors that we're going to work through, right? As Rowan was playing with, we got several different colors, right? This is our control. We have mixed, some weak acid with it, pH of 6, strong acid or stronger acid, pH of 3, a pretty weak base, right? Strong base, right? And some salt water. Now, just so you kind of get an idea, a pH of 6 is 10 times more acidic than neutral, nothing. pH of 8 is 10 times more alkaline or basic. All right, now if we talk about the other two pHs, a pH of 3 is 1,000 times more acidic than 6, 10,000 more times acidic than 7. A pH of 8 is 1,000 times more, pH of 11 is 1,000 times more acidic than 8, and 10,000 times more acidic than 7. All right, these two down here, they are not acidic, they're not basic, they're just some saltwater solutions. So we're going to see how these affect catalase. See that? And by doing this, how we're going to do this is we're going to drop these starch balls that have catalase in them, the enzyme, into a hydrogen peroxide solution. When the enzyme is working, it will break hydrogen peroxide down into oxygen. The oxygen will attach to the spheres and cause the spheres to float. So once they float up, we're going to see how long that takes. The faster it happens, yeah, it's green like grass. The faster that happens, the better the enzyme's working, the more active it is. The longer it takes for that to happen, the more denatured the enzyme, the protein is, and it's not going to work as well. All right, so you ready, Ro?
Yeah.
All right, so we're going to lower this down. I'm going to set these over here, okay? All right, so we're going to do each one of them 2 times, okay, Ro? 2 times. So what we're going to do is we're going to put this right here, all right? We're going to do the pink one first, okay? So, all right, so grab you one. Grab this one.
You try this out.
I'm going to do this one, you use them. All right, so we're going to I'm going to have my stopwatch ready.
Oh.
As soon as I drop it, I'm going to start my stopwatch. So we're going to grab a pink one. Ready?
I want this one.
Okay. We're going to take the pink one.
I can win.
If I can get one. All right, I got a pink one and start. So my timer is rolling. Okay, we're waiting for it. This is our control group. This is our pink one. We're waiting for it to float to the top, right?
Get it. Get it. You help me.
All right, it's floating up, and stop. Okay, so one of our times for pink. Whatever. All right, so one of our times for pink. It's hard to get them, ain't it, Ro? It's 19.32 seconds. We're going to do it one more time with pink. All right, so I'm going to reset my watch. All right, can you get it? I try. Look, you got to squeeze it. You see me squeezing it? All right, you squeeze it, and then you let go. All right.
Hey, on my head. Put water on my head.
All right, so ready? 1, 2, 3. All right, so
I got water.
stopwatch is going. You got it, babe?
Yeah, I got the water.
All right. See it start to rise, start to rise, start to rise, and there. All right, this one took 16.68 seconds.
Daddy, I want chips. I don't want to play these.
You don't want to do these? You want chips?
Yeah.
All right.
I want cheese.
You want Cheetos?
Oh, cheese.
All right, you can't sit right here. You gotta go sit over there. All right, so that's 2 trials. Got 2 times for pink up there. Next one we're gonna do is yellow. Okay? Huh? You gotta go get. I'll go get it in a minute. Okay, you gotta wait. All right, so get our yellow ready. Get our timer going, and start. All right, our timer is rolling. Our yellow starch is in this one here. What's wrong? I'll fix it just a minute. I gotta wait just a second, right? All right, so there is our timer going up and stop. All right, the yellow took 18.86 seconds. All right. Next up, we're gonna do it one more time. Reset. All right. Get me another yellow one. And 1, 2, 3, and start. It's down in here, right? The other one's down there. We gotta wait for it to float. Watch it. Float. Watch. It's gonna start floating in a minute. You see it? There it goes. It's floating, 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 floating. There it goes. All right, 17.71 seconds. Yeah, you see it?
The pink one is up.
17.71 seconds. You on the computer?
Yep.
Right. Let me get our computer. Hang on a second.
Daddy, you get one here. Daddy,
I'll let you lift your chair. All right. Up next. So far we've done pink for 2 times. All right. Yellow, which is just a little more acidic than the pink. So it looks like the times are going to kind of be about the same. So just a slight amount of acid don't look like it's had much effect. Huh? I'll fix it. I'll fix it. I'll fix it. All right, so I don't like the slight acid have much effect. So next we're going to go to purple. This is a very strong acid, right? Or not like very strong. There's 7 stronger acids, but it's a pretty strong acid. 1,000 times stronger than the 6. 10,000 times stronger than 7 or neutral. Okay, so that's the one we're going to do next. Hang on, just get her set up here real quick. You know, just kind of talking. Um, Wait till I have kids till you're ready, because they make everything a little more difficult. But they're awesome. They're a blessing, right? But I couldn't imagine the difficulties of being younger. Right, here you go. Here you go, babe. All right, so what, purple next. So that is, where's my purples? This one, purple, right? So I got my stopwatch ready. Okay, set. So we'll get it ready, and 1, 2, 3. So Oh, I hope I don't miss it. All right, I haven't missed it yet. So here we go. Oh, you can't see, can you? It's in the third one. Here it goes to the top. All right, so 40.68 seconds. So we're going to add that to our right here at purple. So now the strong acid clearly denatured the protein, the catalase, a lot more. So it took it a lot longer to be active. All right, let's do it one more time just to make sure that it wasn't a fluke. And I lost my finger right here. Okay, so I'll put this ready again.